Our last speaker for the session will be, oop, I moved my sheet. Sorry. Dr. Katy Eichinger from the University of Rochester.
Good. So thank you very much for inviting me to speak in this session. Definitely honored to be here with this group of people. Let's see, where do we start? Here we go. So we've heard a little bit about outcome measures, and they're certainly important for clinical care. They are important in documenting how CMT changes over time. Um, well, and they're also obviously important for measuring efficacy in clinical trials. We do have several types of outcome measures that have been developed and validated for use in CMT, and you've heard a little bit about them this morning. The CMT neuropathy score, the CMT pediatric score, the CMT infant score, and then the CMT functional outcome measure. And I'll just a little bit briefly on that one. Originally, it was a 13-item scale that is multi-item in terms of being able to measure strength, hand function, mobility, and balance. So covering lots of different areas of symptoms that are affecting individuals with CMT. And it was scored from 0 to 52 originally, and those numbers are important because it kind of corresponds to some of the data that I'll show later, with 52 being more affected function and 0 being less affected function. We have been able to document natural history in many different types of CMT, but we still have lots of challenges in using these outcome measures in clinical trials. We know that, as you've heard, that many types of CMT are slowly progressive, and these outcome measures aren't quite sensitive enough to pick up those changes. They also just represent a snapshot of function. So this is when individuals come into a clinic space, they perform it, and it depends on how the person's feeling that day and the time and how they're feeling, at that time of day as well. So it can be impacted by other variables. And they also require travel to research centers that have expertise in administering the clinical outcome assessments. So as many other areas of research and technology has really prompted us to consider how to use technology for other potential outcome measures, as well as how to think about different clinical trial designs. And so the use of wearable sensors has really grown with the advances in technology because it's really become much more miniaturized, and we can now fit accelerometers and gyroscopes, which are sensors of motion, onto tiny little chips, making wearable sensors a possibility. We do know from data from big motion analysis labs in CMT that discrete measures of gait and balance may be sensitive to change in slowly progressive conditions. And so when we can use wearable sensors, we can get that same type of data without using large resource-intensive gait labs, which are quite expensive as well. We also have teleconference platforms. So this was really brought to light during the pandemic when we use Zoom and we're able to do some clinical care using teleconference platforms. We also are trying to understand their utility in the research arena as well. That would, again, allow us to capture people from different geographical areas that may not be able to travel to sites. And then also looking at mobile apps, as more people have smartphones, we're able to use mobile apps and the devices to be able to capture data. And again, this does come with some challenges in terms of data privacy laws, enormous amounts of data, and really trying to tease some of that out. But what wearable sensors do allow us to do is look at how people move both in a clinic space as well as in their natural environment, with the natural environment really being the ultimate goal of being able to change function in a person's natural environment. We can use wearable sensors to look at active movement, so we can have a prescribed movement like a walking test and have people wearing sensors and gather data that way. But we can also gather it passively, where they just wear them in their natural environment, their home environment, and be able to capture just passive data as they move throughout their daily life. So when we think about using wearable sensors or digital health technology in general, we need to approach the development of these as outcome measures in the same way that we do with clinical outcome assessments. that does take quite a bit of rigor. We need to have a conceptual framework that backs them up. We need to think about making sure that they're reliable and valid. They're studied in the patient population that you're intending to use them in. And so for individuals with CMT, we know that gait and balance and physical activity are all important features and have been reported to be impacted by individuals with CMT. And so that gives us the conceptual framework to using wearable sensors in this patient population. There are definitely lots of different wearable sensors on the market. You can see here just two different ones that measure movement. And they have, combinations of, like I said, accelerometers, which measure the linear acceleration in the different planes, and then gyroscopes, which measure how the joint angles and how people move. And together, that raw data can be processed using algorithms to give us very small metrics of gait and balance. They can be worn on different parts of the body, and that influences what type of data you're able to collect. So you can see here, one set of sensors is at the chest, the thigh, and the lower leg. That was measuring more physical activity data and position changes. And then the other one on the right-hand side is where the sensors are worn at the waist and the feet, and actually can collect the change in joint angle at the ankle, which is very important for our patients with CMT. Let's see. So, today what I was going to do is just focus on a couple of projects that we have done in CMT to give you an idea of kind of what kind of data we can capture and kind of where we're at in understanding the use of wearable sensors in this patient population. So initially we did complete a pilot project with individuals with, we had 15 individuals with CMT1A and then 15 age-matched, sex-matched controls that wore sensors during the gait and balance assessments of the CMT FOM. So we had them wear them during the 10-meter walk run, the timed up and go, the 6-minute walk test, and different balance tests. And they wore the sensors at the chest, the thigh, and the lower limb. They wore them obviously in clinic, but we also asked them to wear them at home for 24 hours. They were influenced by the battery life, where we could only capture that 24-hour data, but to get a kind of a measure of physical activity in the home environment. And then we partnered with our colleagues in the Department of Engineering to look at trying to create disease-specific algorithms. A lot of algorithms that come with wearable sensors are created using individuals that are not affected by any other conditions. And so you can imagine people with CMT often walk differently than people that are not affected. And so we wanted to try to see if more disease-specific algorithms would be helpful. And so what we found with this study is that we looked at two different variables. We looked at stride, the number of strides, so how many a stride is from like basically heel contact to heel contact of the same foot, how many strides a person took during the 6-minute walk test, and then their stride duration, how long it took them to take that step. And what we found is that people with CMT took fewer strides and had longer duration versus the controls. And then we also want to know, in terms of, kind of validation, is how do our digital outcome measures relate to our clinical outcome measures? And so we looked at the relationship with the CMT FOM and found that stride duration has a moderate correlation to the CMT FOM score at 0.5. But remember that the CMT FOM score, outside of lower extremity strength and mobility, also captures hand function and balance as well. And so those may, influence the correlation that we get there. The other thing that we looked at is we were able to see that short duration of activity. And these are activity clocks, and really the clocks aren't quite as important as the data below. But it's looking at an individual that is age matched with a person that has CMT1A. And what you can see here, we use this again, these sensors were relative positions, so we could tell if a person, what they're doing during their day, looking at lying, sitting, standing, and walking. And there weren't significant differences between the groups, but what you can see is that there were trends that individuals with CMT spent longer time sitting and less time standing than their age-matched controls. So again, just gives us another measure of how people are functioning in their home environment. We are also part of an international project, multi-site project, looking at different sensor systems through the Inherited Neuropathies Consortium. So this was an NIH-funded study led by Josh Burns and Kayla Cornett and myself. And we're looking at both a fairly large population of individuals with CMT, the most common types, 1A, 1B, 2A, and X. And we're looking at how they do using sensors in the clinic as well as measuring physical activity in the home environment. And so they wore the sensors, the in-clinic sensors around their waist and around their feet. And then they took home a physical activity monitor to monitor their physical activity for 7 days following their in-person clinic visit. And so some of the data that you can get from this, and it's, it's a lot of data, and I don't think you can probably see the different variables on the side, but the different variables of gait are listed on the side. And you can see here just how a person with a fairly mild CMT FOM score of 8 compares to someone who has a more moderately affected CMT score of 37. And you can see things like gait speed, which I think is the third one down, where with the mildly affected person has a score of around 100%, whereas the individual with a higher score has a fairly low gait speed. And so we haven't looked at all the aggregate data. We're working on that. But it can give you an idea of how it may be able to differentiate between individuals with different amounts of function based on their disease severity. We also looked at, again, looking at how these discrete variables of gait and balance can relate to the functional outcome measures. And again, here we looked at gait speed on the vertical axis and the CMT FOM score on the horizontal axis. And again, had a moderate correlation in 76 individuals with the CMT1A. And similarly, we can also capture some data on balance. So these, again, are just plots of kind of what the data looks like. And again, there are a lot of different variables that we can measure with balance. This looks at postural sway. So how much is a person moving? We can look at how fast their sway is. We can look at how far their sway is. But there's a lot of different individuals. And again, you can see the individual on the left with a more mild form of CMT versus the person on the right, and just again how that balance may give us some information about how the person is performing from a balance standpoint. And then, like I said, we had them wear their activity monitor home. So now we're looking at how they're performing in their natural environment, what they're doing in their day-to-day life. And this is more passive data collection, and it's we have it worn on their waist. It just, the algorithms pick up the gait a little bit better in that position, although certainly the wrist-worn devices are more preferable to the patient populations or to any of us for that matter. But what it let us look at were variables of step count, and then the amount of time spent in different levels of physical activity. So looking at sedentary activity, light, moderate, vigorous, and very vigorous amounts of activity. It also does capture sleep, but we haven't looked at that data quite yet. Again, you can see here, this is just a single day of activity. And again, looking at someone with less severe CMT versus someone who's more affected, and just the difference in the amount of time they spend in different activity levels. The thing that I would take away from this as well is that the individuals in general spend a fair amount of time in a sedentary amount of activity. And that's important as we think about moving forward. We would always want to try to increase activity levels in this patient population. Again, kind of correlating back to the measures, the clinical outcome assessments, we looked at the average daily steps against the CMT FOM scores, and again, found good relationships. So we're seeing that the digital health technology measures are able to correlate with our clinical outcome assessments. And then lastly, this is a project that we recently started. We partnered with SysNav to examine gait metrics in the natural environment. So this is now looking at some of those discrete measures of gait, but now doing it in the home environment. And this is important because this device and this measure called the 95th percentile of stride velocity has actually now been approved as a primary outcome measure by the EMA. And so if we can all look at how this is working in CMT, again, pick out maybe different variables. Again, these sensors produce a ton of data, and so we may have different variables that are important for individuals with CMT, but we are in the process of studying that through the ACT-CMT study, which is our natural history study in CMT1A. We are, as Mary said earlier, we have extended that study, and we're now adding this sensor into 30 individuals at the University of Rochester and partnering with SysNav to kind of figure out if there are some specific measures of gait and balance that would be important to this patient population that may be more sensitive than some of our clinical outcome assessments and can be used kind of as a biomarker of function, if you will. So in summary, we're definitely trying to use digital technology to develop novel outcome measures, but it does require the same amount of rigor and really taking time to develop them and make sure that we're getting reliable, valid, and sensitive data. They do appear to be feasible in this patient population. People will use them, they will engage with them and things like that, which is really important because the last thing you want to do is hand a sensor to somebody and they take it off and leave it sitting on their countertop. You're not going to get much data from that. So it does seem that the individuals with CMT are welcoming the use of digital technology as we've studied it. And so it is, possible to be able to use these in clinical trials, but we still have more work to do in terms of understanding, sensitivity change. We haven't quite gotten there yet. And so that will be coming as we finish up some of these studies to understand, are they more sensitive than our change in the clinical outcome assessments? And with that, certainly would like to acknowledge the participants and their families. We do appreciate putting stickers on people and wearing devices around waists and things like that aren't always the most comfortable. And so we definitely appreciate everyone's participation in that. My team at the University of Rochester, and then Josh and Kayla. Josh is now at St. Jude, but from the University of Sydney, and the team at University of Iowa that's helped with the INC projects, and then the sites for the INC, the multi-site wearable sensor study. So thank you.
Yeah, okay. I'll ask my own question if that's okay. And then I'll sit down for it. Okay. I'm very excited to see that you're applying this to multiple types. And one of the things that I think Brett highlighted beautifully in his talk is how heterogeneous this disease is. And I'm wondering, in the wearable sensors that you're looking at multiple types, are you seeing any differences between the types or even within the same type?
We haven't started looking at subgroup pieces yet, but that, we need to have a little bit bigger numbers to be able to do that. But that is the hope, that you may see, like we've seen in Duchenne dystrophy, the 95th percentile gait velocity is the most important outcome measure. Well, for one type of CMT, we might pick that out as the most important. And for other types of CMT, again, similar measures, depending on what type of CMT they have and what's affected. So yeah, there's definitely potential to be able to do that.
Hi, over here on the online, yes, in the corner. I have a few questions actually. For translating sensors and real world data, what type of timeline do you need for generating a baseline, like that considers a person's normal activities from week in, week out? How long do you need? Is it 1 day, 1 week, 1 month? And then second would be, is there a way, do you correlate the real world data to the sensors to take into consideration someone at a conference now sitting down all day, it doesn't spike out inactivity?
Yep. Definitely very good question. So the minimal amount of time hasn't fully been determined for each type of sensor. There is some data that would say a minimum of 3 days is important to capture physical activity. I think the second part of your question is important, because I think that, again, pulling certain types of variables out from the sensors is where that's important. So if we look at just daily step count, if you will, yes, we're all sitting here in a conference day. I can guarantee by the end of the day, none of us will have great step counts. So it is influenced by those things, but there are ways to pull out specific variables. So one that we've used in the wearable physical activity one is looking at what we call a max step count. So what is the most steps that a person takes in a 10-second period? And so by doing that, we tease out the variability that happens day-to-day life, weekends versus weekdays. And it also turns, living in the Northeast versus, sunny southern United States where you can be outside all day long. So you can pull out specific variables that are less sensitive to the variability of day-to-day activity.
One last question also,
Yep.
sorry. You said it is a bit of a lot of effort for patients to wear these sensors all the time. Do they get to learn anything along the way about how their disease affects them through these sensors or the real world data?
Yeah. Another good question. So there's challenges there. I mean, I think even just putting on a device and knowing somebody is measuring you can change your behavior. And so we have to think about that. And then if you add the ability to get that feedback, do people again then further change their behavior because of that? So a lot of the ones that are used on a research grade actually don't give that feedback to the participant on a day-to-day basis. But I think there's still utility in that because, I'm doing another study in a different patient population where I'm actually trying to look at changing physical activity from a behavioral health standpoint. And in that situation, I do want them to be able to see it. So I think just like we need to be very careful about what our research question is and the data that we're collecting or the type of device we're using, we also want to look at, again, maybe I want them to have that information. So it depends on really kind of context of use and what your research question is on whether or not you want them to be being mindful of it. But again, there is, a bit of a Hawthorne effect that, people, oh, you're watching how far I'm walking. I'm going to get up and move a little bit more. So, and we just need to build that into our research studies. Yes, we're good.
So before we break for lunch, we have an opportunity to ask questions of all of our panelists generally. So if there's any sort of overarching questions, and I can start with one, which is I was particularly intrigued with the approach that Darius described to take a family of with the heterotetramer, right? And to be able to use natural history data from patients with mutations of 4 different genes. Can we think of any in the field of CMT where that would be applicable?
I would say I'm not a CMT expert, so I'll let the CMT experts speak. But I think for AP4, it's a pretty unique constellation. But one other thought I would add is we know that many, and I think the first Aries talk highlighted this, we know that many of the proteins involved in CMT and HSP, they're at least in the same pathway. So even though they might not be part of the same immediate protein complex, there are proteins that interact directly. So I think maybe extending that concept to a pathway or a group within a pathway, I think could have some merit.
I can't think of any examples that fit that. I think, yeah, I think the subtypes are quite similar as we heard from Mary, but clinically different enough that I don't know of any multiple types that could serve as a control for another. Maybe others have can think of something.
If I could ask another question as well. Okay, this is on now. One of the themes that I think has come up is like outcome measures and using new outcome measures for trial designs. And I'm wondering what those conversations have been like with the FDA during the design process and the execution. How do you include novel outcome measures and novel sensors like you're proposing?
Well, I can piggyback on you. I also have not been involved in those conversations, but I think, you know, being able to see them being accepted in other disease states, is helpful. And there's some, you know, some early work that's being done in different diseases. And I think hopefully we can piggyback on some of that, but, I also have not been part of those conversations so much.
Yeah, I would say for our way of thinking about this, not having had those conversations, sort of preparing for those conversations, I think what we're trying to do is make a case through patient responses about what's most important to them, and then try to tailor our assessments to those specific aspects of the condition, and then also try to anchor those. So, for example, the vocal fold movement. So that's not a measure of a true function that the patient has. So we want to try to then anchor that to something functional, other metrics of voice function or patient-reported outcome measures. We're trying to use an app to capture voice function as well. So we're sort of preparing to give an argument for why these represent something important to patients.
Yeah, I completely agree with this point. I think it's really important for new outcome measures to, in addition to validating them, to show their importance. And I think patient surveys are a great example of attributing importance to what you're measuring. The other, I think, metric that we need to think about is what is the minimal clinically important difference for these outcome measures, and how do we anchor that to patient-reported outcome measures. I think that's something that we need, because just validating a novel outcome measure won't be enough. You have to say, like, what is an important change, and how do you define importance? So I think that's where patient-reported outcome measures can be very helpful.
I have a question over here.
Very nice session. Thanks very much. Would you be able to comment on clinical trial design for all comers, meaning all different CMT subsets, in terms of finding a signal for a therapeutic? Is that a possibility? And would you have any thoughts on the design of that? Thanks.
I think for now, there are efforts to, obviously have been many efforts to develop scales, other biomarkers that are hopefully applicable across multiple forms of CMT. So I think any clinical trial should include the most validated markers that we have, these different scales that we have, and then, but also try to include potentially novel assessments. And I think as Mary really highlighted, we don't have anything that's validated now because we don't have a treatment, so we can't validate a measure as being able to capture a treatment effect because we haven't done that study yet. But every study is a chance to learn, and so the more we can incorporate even into safety trials and efficacy trials, the more we'll learn, and this will be an iterative process. We'll get closer and closer to, I think, a core set of outcome measures that are applicable across multiple forms of CMT. Then I think we also need to be thinking about the differences across different forms and what's important in those forms, and whether there are opportunities to hone in on key features that can be useful for a trial.
A couple of things that I would just add in is we see a fair amount of variability in some of the clinical outcome assessments, and there's definitely lots of different sources of that variability. And so trying to control the things that we can control is really important. Having standardized equipment, having standardized and trained evaluators that conduct them and all the outcome measures in a standardized way, again, just to try to minimize any variability that we can, so that we can see a signal. And then I think, the other piece of that is sometimes, as much as we try and understand the learning effect, we know that when an individual is in a clinical trial and we're measuring over serial amounts of times, that there may be some sort of learning effect on some of the outcome measures. And so at least some of the clinical outcome assessments. And so trying to build that into your clinical trial design at the very beginning, where you may have them perform those assessments at least 2 times before you introduce an intervention, so that you understand the variability within that particular sample, as well as trying to reduce any learning effect as you measure throughout the duration of the study.
Also just add one of the challenges we're having and thinking about the 1B trial in TRPV4 is balancing trying to cast this wide net with the costs and the patient burden. So, we're seeking to hear from patients how much are they, how many visits are they willing to do? How many assessments over visits? They're traveling a long way. How many days does that take? And so that's a obviously something to consider, and also trying to pare down and not make these too burdensome for patients.
Question over here online. So we heard from Mary's talk about the role of the fat fraction MRI and how they're using AI to improve the sensitivity. Where do you think AI can be applied to for an innovative trial design to remove subjectivity and increase reliability?
I do think there are opportunities with some of the digital health technologies because they produce such enormous amounts of data, but you do need to still have enough participants to study where you can have your group that you train the AI on versus the group that you're measuring it in. And so I think there are opportunities because of the amount of data, but that work is still, kind of being thought about, I guess I would say.
As everyone was talking, I was thinking about how much data that is. Do you think the tools for analyzing the data are adequate, or do you think those also need to be built?
I think partnering with a good data scientist helps. And that's really, probably some of my challenges as a clinician researcher is being able to have those partnerships. But they're out there, and we do have some established partnerships that will help manage the amounts of data and really trying to pick out which variables are, again, important to the patient population, are reliable, valid, and by that we'll be able to pare down the amount of data and only look at the more specific things that are showing importance to the patient population.
I think I'll just add, I think the INC has done a wonderful job syncing efforts around the world. I think for HSP, there are consortia in different parts of the world. And I think one way to use AI tools and to build reference data sets is to combine all the available data. And that's not always trivial. So I think syncing and harmonizing assessments across different sites and international studies is really important for rare diseases.
Question. So I think the wearable sensors are really exciting because we're able to have this continuous data and might be able to pick up on some small changes. I was just wondering, are there plans to use this to kind of further optimize the scores, like the CMT FOM? Like, are we seeing data showing that there's certain outcome measures that are variable from patient to patient and might be skewing the data? Are there new endpoints that should be added in? And then kind of to piggyback onto some of the things Brett said, are there further efforts to try to kind of look at CMT subtypes and then kind of optimize the scores again based on that?
Yeah, so we do have quite a bit of data in the INC using the CMT FOM across subtypes, and we do need to put some efforts into kind of analyzing that data. And then we can look again with the INC project where we have the different types of sensors to be able to pull that in. I don't know that, I mean, the CMT FOM itself was rash analyzed, and so I think that that's in a pretty good state right now. I think that, the conversations earlier today about, adding wearable sensor data into clinical outcome assessments may be challenging from a regulatory perspective. But I think there's just more work to be done to better understand how to blend them in. I mean, even when you think about clinical trial design, are there ways to have the clinical outcome assessments, know that they're kind of more of and then have the digital technology where you can maybe more periodically measure somebody's activity in their home environment and reduce the number of times that you need to use the clinical outcome assessments, just to, again, optimize, decrease patient burden and increase, potential signals that you may see. But, it's still, I guess I would say, just still work that needs to be done at this point.
Well, I think it's lunchtime. My belly is rumbling, so probably other people as well. I'd like to thank again our 3 speakers and panelists, and also Dr. Mary Riley again for the great talks this morning.